Hello students, I am RG ma'am is with you to start your journey with the new chapter today. And the new chapter name is Cellular Level of Organization. So students, today we are going to start with the lecture number 1 of Cellular Level of Organization. So before starting the chapter, let me introduce you about the cellular level that has been organized. Means what is a cell? Okay. So first you should know this thing that what is a cell. So it is the basic and structural, or you can say the fundamental unit of a living organism. Now the question arises over here. Is any organism present on the earth that do not contain cells? Yes or no? You should find the answer. I will tell you the answer in the upcoming video, that is lecture number 2. So there is a question in your mind that they may be unicellular also, they may be multicellular also. Means if a particular organism contains only a single cell, then it is considered as unicellular organism. But if an organism has many cells, it may be present in millions, it may be present in billions or trillions. So it is considered as multicellular. So student, what is cell, how the cell was discovered, and how can the cell be seen? Can you see the cell with your naked eyes, students? So this is very important to read about. In this chapter, we are going to explore the cell world. Okay, now I want to tell you about the internal units of the cell. That is the cell organelles, which lies in the cytoplasm part of the cell. Okay, which lies in the matrix. They perform different metabolic activities. We get the ATP, that is in the form of energy, and we perform various functions. So the basic structural and fundamental unit of life is known as cell. So students, before starting this lecture, I am going to show you a very small video clip through which we will understand each and everything. Okay students, so I am going to start firstly with the content part. Okay, so the content part of the first lecture will be, first is the introduction of the cell. So what is cell? Second is cell theory. Who gave the cell theory? How the cell was discovered? How scientists came to know that there is a very small basic unit which is living inside our body. Okay, next is the microscopy, which is very important to understand. Actually, you cannot see the cell with your naked eyes. You need a high power lens, and which is organized through a microscope. Scientists discovered the microscopes, and in nowadays electron microscope is very commonly used to see the little things of a particular cell in a diverse form. Okay, now next is the cell size. Now, there is a question. Are the cell size variable? Yes, students, the cell size are variable depending upon the type of function they perform. Okay, next is the cell shape. Okay, the cell shape. Cells are found in many shapes. They can be spherical, they can be discoidal. Okay. Now, students, I am going to tell you what is cell through a very small video clip, and we are going to understand about the beautiful world of the cell. Okay students, so you are very well known about this thing that cell is the basic structural fundamental unit of life. Now there is a human being and it contains the basic units which is known as cell. Mean its body is made up of these basic units. Okay students, now you are aware with this thing that what are cells. Now the question arises students that in a particular human being, cell resides in millions, in billions or in trillions. That is a good question, and I will give you all the answers of these type of questions because it is a mystery. So students, you have to understand this thing that a body, an organism can contain a single cell also, and it is considered in the category of unicellular. But what about us? Who are we? We are multicellular organisms that contains millions, billions or trillions of cells in our body. Okay, students, so are you understanding this mystery of cells? Now, students, I am going to make you understand about the thing that is very important for this chapter, that cell shape and size may vary. Okay, in our body, these type of cells are present. The neuron is present, RBCs are present, ovum is present in a particular female. But what is the difference? The difference can be in their structure, it can be in their shape, it can be in their size as well. Okay. So these are the basic criteria on which we can divide the cell on the basis of size, on the basis of shape also, and mainly on the basis of functions also, because it contains some cell organelles which perform different type of functions. As I want to give an example over here, like mitochondria, which is also known as powerhouse of the cell. So its function is to perform energy, its function is to produce energy. Okay, so these are the different type of cell organelles like the endoplasmic reticulum, Golgi bodies, vacuoles, and many more. So we are going to understand each and everything in this lecture. Okay, so let's start with the first part, that is about cell. So cell is the smallest structure and fundamental unit of living organisms and is known as cell. Cell word is derived from the Latin word cellula, which means the small room. So you can consider it like that as an example, students, that there is a small room in your house and you have put there a table, a chair, a AC, or many more things or many articles in that. So the things are like cell organelles which are present in a small room, that is cell. So now you are understanding about the definition of the cell, and it is very common. You have been studying it from your very childhood state, that what is cell, what are its function, but we are going to brief each and everything in the upcoming lectures also about what is cell and what are the different cell organelles which are present inside it. Okay. Now, let's move on towards the next part, that is the cell theory. Who gave the cell theory and what was the need for giving the cell theory? So, students, it was proposed for the first time by German botanist M.J. Sheldon in 1838 and British zoologist T. Swan in 1839. So these were both the two scientists, Sheldon and Swan, who proposed the cell theory, and later on it was continued by Rudolf Virchow also. Now, the next part about this thing is that outcomes of the cell theory. Now, what were the outcomes of the cell theory? So the outcomes of the cell theory were: a cell is the basic unit of life. Now, the cell is the basic unit of life. Next is, cells are present in all organisms. So, this is the basic thing. Actually, it is the basic thing of life and it is present in all organisms. Now, the question arises, do all organisms contain cells inside their body? It is a question and you have to find it your answer. Now, moving towards the next slide, we are going to learn about that a cell is the small mass of protoplasm, usually containing a nucleus or a nuclear material along with some organelles. So, what is protoplasm? Actually, it contains the nucleus as well as cytoplasm. So, nucleus and cytoplasm are combinedly known as what? They are known as protoplasm. Next is, cells are physiological unit of life. Now, why I am saying the physiological word over here? Physiological word means that they are the physiological unit of life, that is, they can divide, they can multiply, and they can form new cells also. Next, each cell is capable of maintaining its vitality independently. Now, each cell can survive independently and perform its functions. Okay. Now, you are understanding the world of cell, how it is important to us, and the body, the human body, and I am made up of trillions of cells. Okay. Now, modern cell theory or cell principle or cell doctrine states. Now this is a new world of cell. Actually in the previous time the discoveries were carried on. Okay, and that discoveries were very important for us to understand. So all organisms are made up of cell. That is very common, you know this thing. Next is cell does not arise de novo. De novo spontaneous means cell does not arise spontaneously. Actually what happens, cells arise from pre-existing cells. This is very true. What happens, students, during mitotic division? A parent cell divides to form two daughter cells. Okay, so you can see here I am just drawing a small diagram. This is a parent cell which is going to divide into two daughter cells. So this is a type of division which is known as mitotic division. Okay, now cells arise from pre-existing cells and it was also known as omnicellular e-cellule, added by Rodolphe Virchow. So as I told you, Rodolphe Virchow was the scientist who carried further on the cell theory, and he told about this thing that omnicellular e-cellule means the cells arise from the pre-existing cells. Okay, now the next part, cells contain hereditary material. You can see here is a picture of DNA. The hereditary material, that is DNA, deoxyribose nucleic acid. It is the hereditary material which is contained in a cell. Okay, now we are moving towards the next part, that is about the microscopy. Now what is a microscopy? Why do we need a microscope? Okay, now there are different types of microscope like light microscope, compound microscope, electron microscope as well, which is used nowadays. Okay, commonly. So we are going to study about compound microscope. Okay, so why microscopy is necessary, students? Who discovered the first microscope? How the microscope came into existence and how do we use it? What are the parts of microscope and why do we need a microscope? The basic question arises here. So as I told you, cells cannot be seen with the naked eye. So students, we need a complete compound microscope to see them. Now, we are moving towards first about the early microscope. We are just keeping a look on the ancient time, that what happened in the ancient time. Okay, so first laboratory microscope was developed by Robert Hooke in 1665. So in 1665, Robert Hooke developed this microscope. It was simple and had a combination of magnifying lenses fixed in a tube. So it was just made at that time. So Robert made that time and he was not considered about the newer technologies which are going to be coming in the upcoming world. So he just fixed the lenses with a tube to see some kind of cells. Okay, now another simple but more powerful microscope was built by Leeuwenhoek in 1672. So in 1672 it was discovered by Leeuwenhoek. With the help of his microscope, Leeuwenhoek made many discoveries ahead of his time. Okay, so Leeuwenhoek was the scientist who saw some plant cells under this kind of microscope which he made. Okay, so it was a good discovery and it led to new discoveries for the further time. Now, let us look about the microscope. So, it is an instrument which is used to study those objects that cannot be seen with the naked eye or with the help of hand lens. So, you can see here, this is a sort of hand lens usually preferred in your schools to see a very small thing. If you are seeing a very little word from the hand lens and then when you see it in the broader form. So, microscope was an instrument, or it is an instrument which is used nowadays usually to see each and every thing which is invisible to our naked eyes. Okay, now the bacteria, the viruses or many protozoans cannot be seen with the naked eye. So what we should do for that? We can see in the under the microscope. Okay students, now the next point. A microscope has more than one lens. The first compound microscope was built by F. Janssen and Zacharias Janssen. the Janssen brothers, 1590. So 1590 was the sun when the new discovery took place, and it is still now in use. Okay, you can see in your school laboratories as well, or in the laboratories where the work has been organized for seeing the complete cells, okay, for developing newer technologies, as I told you in the last chapter about the tissue culture also. So what is this? We are just seeing all the things under a microscope, and we are just imagining that they can be like that. Now students, the next point is a microscope has more than one lens. So it is obvious that if you want to see any cell in a broader way, then you have to just adjust the lens power. Okay, now in microscopy, the sample to be viewed is first stained in most cases with a dye and then viewed under the microscope. This is very important point students, because if you are staining, then you can see each and every part of a cell clearly. So to make visible each and every part of a cell, what the scientists or the students do, they first dye it. Okay, they stain it with the safranin or any other kind of dye. Now staining highlights specific regions in the cell, which makes it easier for us to view with its complete details. Now the next point, some commonly used staining dyes are safranin, methylene blue and crystal violet. So these are the commonly used stains which are used to dye a particular cell part and used to make it very clearly visible. Means if you want to highlight any kind of part, if you want to see it, then firstly you have to dye with a particular dye. Okay, so dye used are safranin, methylene blue or crystal violet. Now are you understanding these things? Next is we are going to study about the structure of microscope. Now what is structure of microscope? Do it contains many parts? Yes students, it contains many parts. Okay, so first part is about the eyepiece. What is eyepiece? The lens the viewer looks through to see the specimen. Now what is specimen students? First you have to understand this thing. The slide, the glass slide which you are preparing, you are putting a cell on it and then you are observing under the microscope. This is known as what? The specimen. Okay, now the eyepiece usually contains a 10 into or 15 into power lens. So the eyepiece contains the power lens. You have to remember this thing. Okay, it is adjustable. The power, it can be adjustable. Next is the body tube. It is a head. The body tube connects the eyepiece to the objective lenses. Okay, so body tube connects the eyepiece to the objective lenses. Next is the arm part. This is somewhat bended part which is used to hold the microscope. The arm connects the body tube to the base of the microscope, and we used to carry on the microscope with our hand using the arm part only. Now next, coarse or the rough adjustment. Okay, brings the specimen into general focus. So this is the rough adjustment, or we can say the coarse adjustment. We can adjust it by moving on that circular part, and we want to adjust for looking in a broader way or looking in a smaller way of any particular kind of specimen which we are using to see. Now students, next fifth point is the fine adjustment. Now fine tunes the focus and increases the detail of the specimen, generally used for high power. Now some cells are like that, they cannot be seen easily. If you are staining it also completely, then also you cannot see them clearly. So to watch them clearly, what we do, we just do the fine adjustment of the power lens. Now when it is fine adjusted, what will happen? They will be seen, they will be visible very, very clearly to you. Next is about the nose piece. Okay, a rotating turret that houses the objective lenses. The viewer spins the nose piece to select different objective lenses. So if you want to select different kind of objective lenses to view your specimen, then what have to you do? Then you have to adjust the nose piece as well. Now seventh is about the objective lenses. Now what are the objective lenses which are put in the microscope? One of the most important parts of a compound microscope. Remember this thing student, this is the most important part. If the lenses are not in proper way or they are not adjusted properly, you cannot see the specimen clearly. Okay, that are the lenses closest to the specimen. A standard microscope has 3, 4 or 5 objective lenses as well that can range in power from 4x to 100 also. So you can see that this is a very important part of the microscope. Now students, the 8th part is about the specimen and slide. So what is the specimen and slide? As I told you, the glass slide which is used for preparing the specimen. That is, we are using any kind of cell to see. So what we are doing, we are just cutting it, we are staining it, we are putting the glass cover also over it, then we are just putting it in the microscope, under the microscope. So what is specimen? The specimen is the object being examined. Now if you are examining any kind of cell, like I take an example of cork cell or onion peel, or you are examining any RBC or cheek cells, so these are very commonly seen, and we can see the arrangement of the cells as well. Actually, we want to see the shape of the cell, then this is very clearly seen under the microscope. Most specimens are mounted on slides, flat rectangles of thin glass. Now what are the slides actually? They are just the flat rectangles of a thin glass on which some specimen is put, then it is dyed, and then it is covered with the glass cover slide, and it is adjusted under the microscope, and then it is viewed. So this is the whole procedure which we are going to study further also. Now the specimen is placed on the glass and a cover slip is placed over the specimen. Now you are understanding this whole procedure. This allows the slide to be easily inserted or removed from the microscope. Okay students, now you are understanding each and every part of a microscope is very important. Okay, now it allows the specimen to be labeled, transported. Okay, so first if you are examining any kind of cell, like if you are examining the meristematic tissue or meristematic cell of a plant, then what you will do, first you will examine it, then you will label all the parts of that cell. Okay, now so don't get worried about the microscopy part. Actually the microscopy part is very important in biology. Each and everything can be seen under a microscope that is invisible to our eyes. So it is very important. You have been known about the forensic sciences or the microbiology studies also. It is very common nowadays because of the coronavirus. Also viruses, bacteria, protozoan, fungi, each and everything can be observed under the microscope. Now students, the 9th part, that is the stage part, the platform where the slide is placard. Okay, so the stage is a part where you put the slide over it for viewing under the microscope. Now there are some stage clips also. They are just the metallic clips on which the specimen is kept, that is the glass slide is kept and is being adjusted, fixed. Okay, so these are the metal clips that hold the slide in place. These knobs move the stage left and right or up and down. Okay students, now you are understanding that what happens during the 11th point is about the aperture, the hole in the middle of the stage that allows light from the illuminator to reach the specimen. Okay students, so are you understanding each and everything? That it is very important that the hole is there in the microscope which is known as what? The aperture. Okay, that allows the light to fall on the specimen. Next is about the illumination. Now what is illumination? The light source for a microscope. So what is the light source for a microscope? The light which is coming from a room or for a window, and then it just comes on the concave lens, refracts back, and this illuminator or a condenser helps it for a specimen to be clearly viewed. Okay. Older microscopes used piano concave mirrors to reflect light. So this is about the ancient time. What happened in the ancient time? They use what? The piano concave mirrors to reflect the light on the specimen part. We are moving towards the next part, that is the 13th point, iris diagram. Adjust the amount of light that reaches the specimen. Actually, there is a ray optics behind each and every microscope that works. Okay, what is going to happen? The light is going to come on the concave lens which is fitted there in the microscope, and then it is going to refract back. So when it is refracting back, the main role which is played by the iris diaphragm is to adjust the light which is coming on the specimen. So you can see the specimen completely. Okay, now next is condenser. Gathers and focuses light from the illuminator onto the specimen beam. Okay, now what is the next second task which is very important, which is done by the condenser? It just focuses all the light which is coming on the specimen. Okay, next is the base part. The base supports the microscope and illuminator is located here. So I will tell you where the illuminator will be located and what is base, what is nose piece, what is fine adjustment, each and every part through a very small diagram also and a video also. Okay students, so are you understanding each and everything about the microscopy, that how it is very important? Okay, now we are going to see a very small video clip of the microscope and we are going to understand each and every thing about it. So students, let us see about the microscope, that how does it works. Okay, so you can see each and every part of the cell under the microscope. Maybe it be in higher resolution or maybe it be in low resolution. Okay, now resolving power and the resolution are very important for each and every kind of microscope. Okay, you can see in that in low resolution the picture is blur, but in high resolution the picture is very clearly visible to you. Each and every part is very clearly visible to you. So resolution, and the part which is very important is the light which is falling on it. Now there are the metal clips which are attached to it. These are known as the stage clips as well. Okay, in which we put the specimen over there. Now there is a nose piece and it contains the different kind of lenses. As I told you, they can be in the power of 4x, they can be in the power of 10x, and even in the power of 40x as well. Now this is the eyepiece through which you watch the cell part, each and every part clearly. Okay, now this is the stage part on which the metal clips are attached and the specimen is put. Okay, these are the objective lens you can see here. Okay, now we are putting a specimen on the stage, so we have to adjust the nose piece according to the viewing capacity. Okay, now the coarse adjustment and the fine adjustment plays a greater role over here. You can see here that total magnification used is equal to the eyepiece magnification multiplied by the objective lens magnification, that is multiplied by 10 multiplied by 40, that is multiplied by 400. Now what is magnification? Magnification is equal to the size of the image upon the size of the real object. So you can calculate the magnification by applying this formula over there. Okay, now each and every part of the cell can be visible under the microscope clearly when you are staining it properly and putting it under the right power lens in which you can see it clearly. Okay, so you can do the calculations of the magnifications as well. So this is the whole procedure. You can see here the specimen is put over here, the light comes under it and then it refracts back to the concave mirror. The illuminator plays a greater role and the condenser plays a greater role, and you can see each and every part of the cell. Okay, so cell may be visible under the microscope clearly when you are preparing a slide which is impurity free. You are staining it properly, putting the glass cover slide over it properly. Now you have understood about the microscopy very clearly. Okay, now we are going to revise each and every part which we have read in the last slides. So first we read about the body tube. So this is the body tube. Okay, you can see here this part is considered as what? The body tube. This is the eyepiece. So the person or the scientist or the student which is going to observe this specimen put an eye over it and see the specimen. Okay, next is the revolving nose piece. Now why we are using the revolving word here? As I said, you can adjust the nose piece according to the power lens. So these are the different types of power lenses which are used here. So we can adjust according to our specimen. Next is the objectives which are used as power lenses. Now next part is the stage clips. This is the stage part. You can see here this broader part flattened part is known as stage, in which the two metal clips are attached and the specimen is put over there for viewing. Next is the diaphragm part. This is the diaphragm part which is present just below the stage clips. Next is the light source. Very important. The light source, this is a concave mirror which is attached here. Now when the light falls on the concave mirror, it is just reflected back to the specimen part. Okay. Now it is the arm which I told you. This is the arm. Okay. We used to hold the microscope through the arm part only. Next is the stage part, as I told you about the stage part. Coarse adjustment and fine adjustment. Now there are very two important adjustment. In coarse adjustment, what we do, we just put the knob and we just revolve it and see the specimen. But in the fine adjustment, if you want to see each and every part clearly and if you want to make it visible, then you have to adjust the fine adjustment part. So this is the knob which is to be adjusted. Next is the base part. This is the base part of the microscope. Now this is the base part and it holds all the weight, all the things of the complete microscope. Now students, we learned about the microscopy, what is microscopy and why it is important for us. Each and every human being which is studying about the science need a compound microscope or an electron microscope to make each and everything visible under it. So if you are doing any kind of studies which are related to the invisible things, then you should consider a microscope there, because each and every cell part can be seen under the microscope clearly. But it is very important, students, for you all to understand that a specimen should be prepared clearly by staining procedure. The staining procedure may include the complete dyes like the methyl blue, or as I said about the saffronin or the crystal violet there. Actually, it highlights the part which we want to see under the microscope and make them very clear in vision. Okay, so it is very important that you should make a complete study about the microscope and understand each and every part of the microscope as well. Okay, so now I am going to tell you about each and every part of the microscope which we talked in the thorough portion in the last slides. Okay, about the nose piece, the aperture part, the arm part, coarse adjustment, fine adjustment, and very importantly, the light source. Actually, if you are putting the microscope under the diagram, then under the diagram you cannot see any kind of specimen, you cannot see any kind of visibility under it, because a microscope needs a complete lighting source, and a light source is very necessary to be focused on the concave mirror which is attached on the base part of the microscope. So it is very necessary that if you are viewing any kind of specimen under it, then you have to keep the microscope near the window or the gate or the place where the complete light is coming there. So all these parts are very necessary. If any kind of part under the microscope is seen, then it can be labeled as well. And students, each and every part of the microscope is very necessary. So take care of it and keep the microscope in a safe place. No part should be broken down, because if some part get damages, then there is a very problematic condition. Then how you can observe the thing which you want to view under it. So this is very important that each and every part should be maintained. The cleanliness of the microscope also should be maintained. The power lenses should be cleaned every time. Okay, the objectives should be cleaned, and the light source concave mirror should also be cleaned, and even the stage clips which we are using should be fixed. Okay, the specimen which we are keeping on it should not move here and there during viewing under it. Okay, so we are end over the microscope. Next we are moving towards the next slide, that is about the cell size. Now what is cell size, students? Are you confused about any kind of cell size, or you are knowing it better? So what is a cell size? So cell size is, cell differs in sizes in different kind of organisms as well. So cell size may differ from very large and ostrich egg from a very small size, that is sperm cell also. So let us take a look here. Size of cell vary from very small cells of bacteria 0.2 to 0.5 mm to the very large egg of ostrich. The size of the cell is related to its functions. Now, the size of the cell is just and just related to its functions they perform. Okay, now we are going to take a quick view about the cell size. The smallest cell, that is the pleuropneumonia-like organisms, PPLO, also known as Mycoplasma, Gallisepticum. Pleuropneumonia-like organisms, that is 0.1 to 0.5 mm in size variation. So you are seeing this is a great variation in size. Okay, pleuropneumonia-like organisms, that is PPLO, are the smallest cells, okay, present on our earth. Next is largest animal cell. So the largest animal cell is ostrich egg, which ranges to 170 mm to 130 mm in size. That is, it is 18 centimeter in diameter with shell and 15 centimeter without shell. So if you consider it with shell, that is in 18 centimeter in diameter, and if we consider it in without shell, then it is a 15 centimeter in diameter. Now, the most important point, longest animal cell, that is the nerve cell, which is 90 to 100 centimeter in length. The nerve cell, which is very important cell of a human body, the neuron, which ranges between the size 90 to 100 centimeter in length. So these are the various great sizes in which the cell vary. Now, you can see here that the largest plant cell is Acetabularia. You can see here in this picture also, this is what? Acetabularia. Okay, now there is a mystery that what these two pictures represent. Okay, so we are going to solve this mystery. Longest plant cell, that is hemp fiber. So these are the hemp fibers. Okay, so these fibers are used in making different kind of fabric materials also. So these are the longest plant cell. Okay, so there is a difference between the largest and the longest. So remember this thing, students. Okay, so largest is Acetabularia and longest is the hemp fiber. Now, if we talk about humans, largest human cell is the egg cell. Okay, and the smallest human cell is the sperm cell. Okay, so the largest human cell is the egg cell and the smallest human cell is the sperm cell. Now, students, we are moving towards the next part, that is about the cell shape. So, as I told you about the cell shape also, cell shape may vary. They may be spherical, they may be rod-like, they may be discoidal. Each and every shape they can take. Now, students, are you thinking that our body contains different type of shapes as well in cells? Now, put your brains over here that I told you in the past video that some cells differ in shape greatly, okay, but they perform different kind of functions as well. Now, if you are talking about the neuron, that is elongated in shape, okay, the RBCs which are discoidal in shape, the columnar cells are present inside our body, flattened cells which are present on our skin on the body. So, the shape may vary from different parts of our body, as I told you. Actually, our body is being organized by God as well. So, all the cells are different in a particular type of organ in our body. So, neuron, which is elongated in shape, as I told you. Okay, now different cells like muscles. Okay, you go to gym and make muscles. So, which kind of cells are present inside your muscular part? That is the spindle-shaped cells, which are present in the muscular part of your body. So, the ovum which is present inside a female are considered as a spherical in shape. So, these are the different type of shapes which are present around your world, but you cannot see them because you have just learned about the definition that cell is the fundamental unit of life. But the shape may vary from one organism to another organism as well. Okay, different type of cells are present in the different type of organisms depending upon their type of skin, the texture, hair, fur, each and every type which is present in them. So, you can consider that the each and every part of our body, like the nails we have, okay, the hair we have, eyes we have, nose, each and every part of the body consists of different kinds of cells in it. Okay, so you should be aware, and I will make you aware in this cell shape part, that your body can contain different type of the shape of cells, okay, like the flattened cells, columnar, or the spindle shaped, the discoidal shaped, spherical shaped, and many more shapes we are going to discover in this part. Okay, so we are going to study about the cell shape. So cell shows variability with respect to their shape and size in multicellular organisms. So in a multicellular organisms, what happens, students? The cell shows variability with respect to the size as well as shape. Now, shape of the cell may be variable, that is constantly changing. So they can be changing constantly. That is, amoeba and leukocytes are fixed. So amoeba can change its shape as well. They can be in any kind of shape they want to take. Okay, so amoeba are very flexible. They can change their shape, as well as leukocytes also. Now, next part is about the shape of the cell is usually related to its function. So the shape is usually related to its function. Now we are going to study about the different type of shapes of cells. Okay, students, so there are very different type of shapes. So first is flattened. Okay, so example, it is present in skin cells, that is the upper layer. So you can consider the upper layer of your skin. So it is flattened shaped cells which are residing under our skin. Next is the columnar shaped cells. Cells lining the intestinal part. So the cells which are lining the intestinal part are columnar. Next is discoidal, example RBC. RBCs are the discoidal in shape. You can see here, this is like disc. Okay. Next is spherical in shape. Example, eggs of many animals. So the eggs of many animals are spherical in shape. Okay. Now, spindle shaped, the example, smooth muscle fibers. So smooth muscle fibers are spindle in shape. Now what is spindle shape? This type of shape is known as what? The spindle shape. Okay. This is the spindle shape. Okay, now next is elongated, example, nerve cells. Nerve cell, as I told you, the largest cell which is present in a human body are the nerve cells, and they are what? They are elongated in shape. Next is branched, example, pigment cells of the skin. So the pigmented cells, the melanocytes which are present in under the skin are branched in shape. Next is the bean shaped, example, guard cells of stomata, melanocytes. So bean shaped are the guard cells of stomata. So you can see here, this is a cartoon picture of guard cell. So this is bean shaped. This is a bean shaped. Okay, you eat rajma as well. So it is bean in shape, the kidney shaped. You can say that. Okay, now we are going to explore each and every diagram which we have learned about the shape of the cells. So first is the columnar shell, which are found in the lining of the intestine. Next is the flattened cells, which are found in the skin cells, that is the upper layer. So you can see here, these are flattened in shape. Okay, these are flattened in shape. Okay, now next is discoidal cells and spherical cells. So you can see here, these are disc-like, they are discoidal in shape, and they are spherical. This is the ovum. Okay, this is the ovum which is present only in females. So they are spherical in shape. Okay, next is spindle shaped and elongated shape. So as I told you, this is a spindle shape which is present in the muscular muscles. Okay, which is the muscular cells, and next is the elongated shape. This is the nerve cell. Okay, the nerve cell which is present in the human body, which is the largest cell of a human body. Next is the branched shaped and bean shaped. So this is the branched shape. Okay, so these you can see that there are branches of these cells. Okay, next is the bean shaped, the guard cells. These are the guard cell. You can see here, they have a bean-shaped structure which is present in the stomata, which helps in the stomatal opening as well. Now students, we are over with the lecture number 1, that we talked about what is cell, what is the world of cell. Now we are dwelling into the world of cell and we are going to explore new things in this particular chapter. So we will meet in the next lecture, that is the lecture number 2, but let us revise quickly before that what we read in the lecture number 1. So we studied about what is cell. Cell word has been derived from the Latin word cellula, which means what? A small room. Now we read about the cell theory, which was given by Sheldon and Schwann, and which was again continued by the Rudolf Virchow, and according to it, the omnis cellula is cellular. That is, the cells arise from the pre-existing cells. Okay. Now, we talked about the microscopy. Why microscopy is important? Actually, microscopy is very important in today's world as well as it was important in ancient world. But the discoveries took place now and we can see each and every part which is invisible under the microscope because it makes it visible. Okay, we can adjust the coarse adjustment through fine adjustment. Okay, the lenses which are used over it. Okay, we can adjust the nose piece to see each and every part of a particular cell. Okay, so microscopy is very important in today's world. The microbiology is the very biggest branch because it is meant for the research work as well. Okay, now we read about the cell size. What are the cell sizes? It may vary from a very small cell to a very large cell. So a very small cell counts for the PPLOs, that is the pleuropneumonia-like organisms, and which are also known as mycoplasma, and the largest cell is the ostrich egg, which ranges between the 18 diameter. With shell and without shell, it is 15 centimeter in diameter. So it is a very large in size. Okay. Now we read about the cell shapes as well. What are the cell shapes? They may vary from columnar to elongated, discoidal, spherical, spindle shaped as well. So these are the cell shapes. Now in the next lecture we are going to talk about various kind of cells, that is, they may be unicellular, they may be multicellular. Okay. They may be prokaryotic, they may be eukaryotic, and about the plant and the animal cell as well. So students, we are leaving now and we will meet in the lecture number 2. Thank you.